Many thanks. Yes, after discussing with the organizers, we thought to bring another story about codon. I hope I can explain this in a amount of time that is allowed. So yeah, thank you for the very nice introduction and the invitation to talk here. My focus in this talk primarily is about rare diseases, and rare diseases are diseases that are defined as diseases that affect less than 1 in 2,000 individuals. But by their large numbers, they actually affect a very large fraction of the population. It's estimated that more than 30 million people in Europe are affected with a rare disorder. Not all, but often, these rare disorders have a genetic cause, and it's of prime importance for the patients and their families to pinpoint these genetic causes. And this is because once you identify the genetic basis of a rare disorder, you can this allows to perform genetic testing, so identify potential family members that are carriers of pathogenic alleles. We can detect couples that are at risk to get children affected. These couples can perform pre-implantation genetic screening. The families can connect with each other and the clinicians following those families as well. And because we here have a clear root cause of the disorder, we also have a rationale for therapy. However, it remains a hot topic to have proper tools to identify those causes. Traditionally, one will naturally look at coding sequences and variants that affect the proper coding sequence, such as stop codon or frame shift, are obvious candidates for giving the loss of function of a gene, but there are also genes outside the coding sequences. And one of the primary categories are variants that affect the structure of a messenger RNA. And among them, variants that affect splicing are one of these major causes of this type of rare disorders. And this has been identified early on, back in 2005, and we still see maybe 20 to 30% of the diagnosis as of today are based on variants that affect actually the splicing of genes. However, predicting just from sequence alone which variant could affect splicing is non-trivial, and the reason is that the genetic underpinning of splicing is complex. So splicing is the process of removing introns of an RNA, and beyond these canonical dinucleotides, which are determining the boundaries of introns, so the acceptor site here and the donor site over there, there's a lot of sequences that also are important for the determination of splicing, including this so-called branch point over here, this A here, and also sequences that are flanking those positions. There are enhancers and repressors of splicing that are located in the intron, but also in the exons overlapping with the coding sequence. So this entire code is still complex and still a subject of study. So we have started to look into this around 2018 with a talented PhD student, Jun Cheng, in the lab, who we started to look at this using deep learning models. And the idea there was to model with deep neural networks these different regions that I just mentioned to you in the slide before. So the model called MM-Splice is a modular model that models these different aspects of splicing. There's a module for the intronic region, a module that is centered on the acceptor site, another on the donor site, convolutional neural networks on the exon, and also another one on the intron on the 5' side. And so those modules have been trained jointly to predict different aspects of splicing. And there we've been using very large data. One source of data where the actual annotations according to gene code of donors and acceptor sites. So there's over 250,000 of those, as well as perturbation data from a massively parallel reporter assay, where the lab of Georg Selig has generated over 2 million sequences looking into key regions that can affect splicing. And these data were very useful to predict the effect of variants that are, for instance, not seen in the normal population. So at the time when we were developing this model, there was a challenge, the CACHI-5 challenge. And the CACHI-5 challenge was set up like this. So the organizers of the challenge have generated mini genes in which you had in a cassette exon two variants, the wild type variant of the cassette exon and an alternative one, including a variant. And for each of these two mini genes, a measure is done, which is called the percent splicing. That is the percentage of the RNA of the RNAs that contain the cassette exon. It's called psi. And so you have a psi for the reference, the psi ref, and a psi for the alternative, and you can compute the difference, delta psi, which is the difference between two fractions, two percentages. And these were given for half of the library, about 1,000 variants, and the other half was hidden to the challengers, and it was the goal of all of us to predict those. However, we felt that this was a very hard problem, because if I have a variant that has a very strong effect of splicing, that you could, let's say, abolish splicing, and I start from an initial level psi ref, which is very strong, 100%, then I may predict a delta psi of 100%. But if this reference level is low, it's at 10%, then I would predict 10%. If you don't tell me what the psi ref is, I may be good in predicting what are strong variants, but I can fail on that metric, the delta psi. So we contacted them, and they decided to change the challenge and provide delta psi ref for everybody. And for us, we knew also slightly a bit more from observations in data. Show you here on the left-hand side data from GTEx. You have two times the same individuals, a control individual and an individual that has a rare variant near a splice site here close to this exon number 7. And this is for two tissues, the brain tissue and the lung tissue. And what you see in the control individual that has just reference sequence, psi to start with is 70%, and here it's 11%. And there is what I was telling you is that the effect of these variants is much stronger when you start from higher than from lower. If you do very simple biophysics and you have very simple a model of how a variant affects fold change between isoforms, then you go back to your classical scaling law and you can transform this into an effect in terms of proportion of isoforms. This is just simple sigmoid that you have in any logistic regression. And so what we were seeing in the data is often these variants have an effect which is just constant on this logit scale, but by acting on different psi ref have a different effect on the proportions. This was extremely well shown by a paper from Bedliner that dissected in an entire gene with 9 different variants and showed exactly that the scaling laws, up to the very few exceptions of an interactions, actually explain most of the nonlinearities we see in splicing. So we, as I said, the challenge organizers changed the challenge, provided psi ref, and then we were lucky to win this challenge. I want to show you here the scatter plot of our predictions over the observations. You see that most of the variants were not doing much, and some were having strong effects. One could say that this is what is driving the entire correlation. That is correct, but if you think of people with rare disease, they are exactly in this corner, and this is where you want also the predictions to be correct. So we think this is maybe not too bad. And then we continued and applied this to analyze genomes of individuals. Now, you may have heard from many other talks, for example, from the Rex Viscosi, this field of interpreting variants with better tools and better measures that we do on regulation is improving a lot. But still, we can't interpret most of the variants. And as a matter of fact, as of today, when an individual suspected to be affected by a rare disorder undergoes a whole genome sequencing, in half of the case, there's no diagnostic established. We see the variants, potentially we see the causal variants, but we are not able to interpret them and to have a confident prediction of the effects they have. So this is a problem we've been working on together with the colleague Holger Prokisch, who is also at the TUM at the Helmont Center. And for several years now, we've argued that if we can't conclude from genome, we should complement the data with omics data from the patient. And we have a series of work on RNA sequencing based on that. So we complement DNA with observations made on RNAs of the patient. And this is the first paper we did together. We looked here at patients that are suspected to be affected, to have a mitochondrial disorder. You see this on the biochemistry of the biopsies of the muscles of the patient. And in these projects, we look at 100 of those individuals. Half of them could be diagnosed by looking at the exomes. Another 50% were not diagnosed. And from the patient fibroblasts, we perform transcriptome. And since we are here in a rare disease case, we've been looking at anomalies in the transcriptomes, aberrant events. And these were aberrantly expressed genes, so genes whose expression is outside the physiological range, or aberrantly spliced genes, or rare variants that turn out to be monolithically expressed in those patients. And for this very particular disease, we're in a lucky case where directly on the fibroblasts we can perform assays to prove the causality of our candidates, either by supplementation assay or by complementation assays. And in this case, it allowed us to establish 5 new diagnostics. And so in the context of this presentation, I'm going to show you what we do bioinformatically to call aberrant splicing here. And this is a tool we've developed a bit later called Fraser. Well, as I said, the concept in rare disease is to look for anomalies. And so in this case, from a data science point of view, outliers, so those data points that usually we want to remove because they annoy us, they are the actual things of interest. So one can reuse with an inverse purpose these anomaly detections for actually finding interesting patterns. And so here we use a concept that is quite well developed and that I know also quite well known also in bioinformatics, called denoising autoencoders. So denoising autoencoder is a way for you to detect outlier in data. And the concept is simple. You start from your data. So here this is the X. And what you do in silico, in a computer, you corrupt this data. The kind of noise model that you expect is maybe of the type of outliers you would see in your real data. And now you turn the problem into simple supervised problem. You give to your algorithm the corrupted data, and the task is to retrieve the original one. And the difference will be the noise. So we just borrowed this concept, applied it to RNA sequencing. We've done it for expression level. Today I show you for splicing. So this is work that Ines Scheller and Christian Mertens in the lab have done in this tool called Fraser. And so basically it's the same idea, except that the loss is not the one I showed you before. That was a least square regression. We use a loss for these fractions of counts. So it's the beta binomial regression that we use on the output. But otherwise, it's the same idea. You map your data into a latent space that is smaller, so you have the bottleneck there, and that learns a lot of the covariations that we actually see. And we see a lot of covariations in splicing between the individuals. And we can control for that, but we see the contrast between the observation and the typical covariations that we see are those outliers. We can benchmark that, not just by doing these in silico noise injections, but also by comparing to orthogonal data. So in the case of those individuals, we also have the genomes. And so we can see whether we see good enrichment among our outlier calls for positions where there is a variant that is predicted by tools like MMSplice and others for affecting splicing. And the scatter plots on the left-hand side just show you that our methods outperform alternative methods that do this at different levels of significance. So here's an example, a concrete example of what you find with Fraser. What you see at the top are 2 individuals. And you can appreciate here that these 2 individuals show splicing of an exon here that is not annotated in the reference genome. And the 2 individuals that I show at the bottom are individuals that also are affected by this disease, but not because of this gene. There's no problem for them on these positions here. So what happens here is that there's a rare variant that is deep intronic. It's pretty hard for tools to predict that these rare variants will cause splicing. Cause a splicing defect. And this exon contains an in-frame stop, and then this leads TMMDC1, an assembly factor for complex 1, to not be functional in those cells. And complex 1 is important for cellular respiration, and this is the cause of this disease for those two individuals. Now, what Holger spotted very nicely, you looked at the data, said, Julien, look there, you do have also a bit of splicing in these people that are not affected. And actually, that's something probably everyone in the room also has here. This is a place in the genome where either it's an ancient exon or there's a future exon, there's something that looks like an exon. It's not very strong, but it gets affected by a single variant. It gets activated by a single variant. If you think of the scaling law, like we are not fully in the plateau, we're maybe at the beginning of where the scaling law is increasing. And so this is where a little jump in the delta log can actually give you this activation. So that tells us also that we need those positions to be annotated to really interpret variants. Okay. So in this study, we found that 80% of what we call private exons were actually of this kind. There was weak splicing in the other individuals. So we developed further those tools. We looked at way more data. We don't only look at aberrant splicing, also look at aberrant expression and monadic expression. We have several collaborations on this and many users. And by now, we see this increase the diagnostic rate for by 15%. So typically 15% of the samples that were not diagnosed from exomes and then go through RNA sequencing can be revisited and help finding diagnostics. So one critique or one concern that many people have with this strategy, you see, we are starting from fibroblasts, and one critique is that many diseases are actually not manifested in samples in tissues that you can easily biopsy. So if you have patients that have a problem in brain, it's not very clear whether you will open up the brain and extract cells to perform such an RNA sequencing. And therefore, there's quite a strong motivation to be able to predict aberrant splicing in clinically non-accessible tissues. And this is something that Niels and Hassan have been looking into a study that we recently published called developing a tool called AB-Splice, where the goal is to predict aberrant splicing in any human tissue. So here I mentioned you that we have developed Fraser. So this is this tool that's called aberrant splicing, and we could apply it to the GTEx dataset. And the GTEx dataset is an extremely large dataset with about 1,000 individuals. And we have biopsies postmortem for those individuals of about 50 tissues. And for those, we can call the variants. We have 8 million rare variants within gene boundaries, and we've called with Fraser the aberrant splicing events. So this gives us a ground truth across many tissues, accessible or non-accessible usually, for benchmarking tools that predict aberrant splicing. And so when we run MM-Splice and Splice AI, which is a deep learning algorithm from Illumina that predicts creation or loss of splice sites, at this benchmark, we see that both are performing poorly. We are even poorer than Splice AI. So if you focus here on this precision recall curve at 20% recall, and then Splice is below 10%. Splice is slightly above 10%, but these are numbers that are not very great for diagnostics. So we wanted to improve this, and we had these lessons about the fact that we need a reference site for MM-Splice to really work. So Hassan went through the entire GTEx and annotated all the splice sites, even those that are not in the standard annotation, get a quantitative level down to 1% for any tissue. And that has been very helpful. This is helpful because it can tell you whether a variant is simply on an expressed gene in this tissue, or nearby an expressed exon for that tissue, or nearby one of these weak splice sites. And when you combine all that, then you can boost MM-Splice from below 10% to nearly 20%, so you get a twofold improvement of our tool. And now if you combine MM-Splice with Splice AI that predicts creation or loss of splice sites, we get a splice DNA that is about 30% precision and 20% recall. So we get this threefold increase with these splice maps. Now you would say, just as we said with Holger, like if you cannot predict, why don't you measure? We can't measure in brain, but we can measure things from fibroblasts or from blood. So if we use these accessible tissues, maybe we can improve further this tool. And that's what we've done there. We call the aberrant splicing in several tissues. It turns out fibroblasts is the best predictor. And so it gives you, just by itself, a pretty good precision recall curve, but it stops at some point pretty abruptly, and this is where you have splice sites or genes that are just not common between those two tissues. But if you combine them with DNA-based predictions, you get this brown curve here, and now at 20% recall, we are reaching 60% precision. So we have a six-fold improvement in precision in this area, and globally a pretty large improvement with this technique. So the lessons of this aberrant splicing work is that, well, on the one hand, in terms of maybe a bit of the biophysics underlying splicing, is the scaling law is important, and that's an argument also to have maps of reference splicing that combined with predictors of differential effects allows you to boost sequence-based models, and then combining this with a measurement from patients in accessible tissue can even further improve. So there are future works on this field. We can look at things at the protein level, not just at the RNA level. We can predict about expression, not just about splicing, and this is ongoing work. How am I on this time? I'm good. Okay. So in the second part of this talk, I want to switch gears a bit and go into more basic biology. And that's a study from Pedro, a PhD student in my lab, who looked at cellular energy and its relation to codons. As we heard a bit earlier today, the genetic code is redundant for every amino acid. You have multiple ways of encoding it through codons. And as we also heard, there is also different effects of codons. So some codons are more easily decoded. They have a pretty fast decoding time compared to others, and that leads to also higher translation. What is a bit less known is that codons that are rapidly decoded or codons that are slowly decoded lead to degradation more easily of RNA. So there's a coupling between translation rate and degradation rates of RNAs. So this leads the question is whether this can be regulated, and that has been a hot topic of research. Are codon effects regulated? One of the logical way codon effects could be regulated is the tRNA, so the molecules that bring the amino acid and are decoded every codon, if their abundance are themselves regulated. And there's some evidence, but a lot also of evidence that actually the pool of anticodons is pretty stable between tissues. So maybe there's no regulations of codon effects, and if so, then maybe there's something else than cognate tRNA abundance. So to study that, we looked at GTEx. I just mentioned to you this data set. GTEx is steady-state RNA. We can't really look at half-life there, but we can use a trick that is often used in bioinformatics, and that's a trick of looking at the coverage of your gene in terms of exonic and intronic coverage. And the idea is that the precursor RNA, which contains the introns, has a coverage that kind of captures your RNA synthesis rate, whereas the coverage of the exon captures the steady state of the major RNA. And so if you look at the ratios of the two, you have a kind of Postman metric of the inverse of the degradations of the stability of your RNA. The bigger the proportion of exonic rate over intronic rate is, the more stable is your RNA. So there's many biases in this metric, but we're looking at for every gene at the variations of that between individuals and between tissues. So I told you that codon usage have an effect on half-life. So what I'm going to look at now is how this metric here associates with which codon I find in my gene. So far, so good? Okay. So the first one, I think, is a CUG, and that's a rapidly decoded codon. And we look here at different bins. I'm just binning the frequencies of these rapidly decoded codons across RNAs. And for each bin, we look at this relative RNA half-life. So here you have all the genes that have nearly 10% of their codons are CUGs. And what you see is there's no big difference in terms of this exon-intron coverage, so in terms of their half-life, probably, between brain and skin. Whereas if I go down to here, to genes that have rarely using these rapidly decoded codons, there's a very strong difference. So they seem to be relatively more stable in brain than in skin when they use this, when they don't use the rapidly decoded codon. So this is for one codon, but what you can do is you can get a metric that works for the entire RNA. And so what I'm going to use now is a metric that we could borrow from Tagliatella that computes the average decoding time of an mRNA. So this is what I'm plotting here on the x-axis. So that's the mean reference decoding rate. Yeah, if you say the average decoding rates of all the codons in an RNA. And on the y-axis, I look at this fold change between the skin and the average tissue. So in a way, it's a reminiscent of this pink curve here, but using now plotting all the genes. That's why you have this huge scatter plot, not doing any binning, and looking at on the x-axis across all codons. And what you see is a trend. So there is a kind of dynamic range of about 1 codon per second in terms of decoding time between genes. And these associate with about 1.6-fold fold change in terms of half-life. So that's quite strong. It's nearly a 2-fold change just across genes, just explained or associated with codon usage. And that's for skin. And you saw for brain, it was going the other way around. So what we compute then is what we call the Comedy coefficient, for codon optimality mediated degradation, and that's the slope of this curve. So this captures how important it is to be optimally encoded or not in a tissue. So if it's strong like this, it is important. What we have now is this coefficient is changing a lot between tissues. It starts from 0.7 and goes all the way to 1.5, with brain having the lower association and skin having the stronger association. So we have something that varies between tissue. We have now a metric, and we can ask, is there a basis? Is there something that drives this metric? I can compute this slope, not just between for a tissue, I can compute in any sample of GTEx. Okay, so far so good. So we do that, and then what we see is that we've done some gene ontology enrichment and so on. I'll just show you here for one specific gene of mitochondrial pathways. So this is a mitochondrial gene that is whose expression is actually associated with this Comedy coefficient. So the higher the mitochondria, the lower the Comedy coefficient. That corresponds also for tissues where you have high energy, like in brain or muscle. If you look with age, we also see an association. And so older individuals have a stronger Comedy coefficient. So there's a stronger codon effect in older individuals than in younger individuals. And that's also related to energy metabolism. With age, energy metabolism is reduced. And that gives us also, again, this association that codon effect seems to be more important there. So Pedro went and tried to explain that with a biophysical model. As you may know, translation is for the cell a process that is highly energy demanding. And so what he's been doing here with a mathematical model is to relate the abundance of ATP and GTP, which are these two energy molecules that are in rapid conversion to each other, to the loading ratio of the ternary complex, which is the complex that contains the loaded tRNA. And what he found is that if you have low ATP, then you have a very strong difference in terms of how fast you will load a fast codon over a slow codon, and this gets much more decreased when ATP increases. So this is from a biophysical modeling using reactions that are quite common across eukaryotes, a possible explanation why we would have in high energy less differences between optimal over non-optimal codon, because we'll have less differences in terms of ternary complex loading ratio. So all this is for now correlations or mathematics. We needed to test that. And we looked at the GTEx again. And in GTEx, we have a postmortem samples. So what happens is the samples have been collected after death. And after death, you don't have oxygen coming to tissues. So we have the first perturbation where we're not bringing oxygen to the cells. And the sampling time, you can imagine, it's tough to do those experiments, have been different from individuals to individuals. So what we can do then is we can correlate the ischemic time to this Combi coefficient. I'm adjusting here for age, and we see this correlation. So we see again that if you deprive the oxygen, so you have less ATP probably, you have a stronger effect of stronger differences between optimal and non-optimal codons. And if you don't like this adjustment and these linear things, I can show you more the raw data. This is one tissue. Everything is stratified by age. And you have the ischemic time on the x-axis and this Combi coefficient. And we have, again, this slope very clearly. So, of course, this is a lot happens after death, and it might be confounded by other factors. So we needed something potentially more fast, but we had to go to yeast for that, and we collaborated with Vicente Pellicciano, who's developed an assay called 5p-seq that allows you to very rapidly map where ribosomes are on an RNA. And in yeast, there are data that shows that when you put anti-lysine A, you can reduce, and we know by how much, the intracellular ATP concentration. And what I show you here is that the occupancies of slow over fast over slow codons really change according to ATP concentration. And so we have a big gap when the ATP concentration is lower. This also summarized with this scatter plot here. And that is consistent with our model that in low ATP concentration, there's a higher ratio of loading of the TC, of the ternary complex, and therefore lower occupancy of the ribosomes on those codons. So to conclude, we think this data shows that cellular energy levels, in terms of ATP or GTP elements, provide a new coupling between energy metabolism, gene regulation, and codon usage, and that this coupling is independent of tRNA regulation. All this is assumed, assuming constant tRNA abundance, and that is consistent also with several measures from recent publications. So this analysis explains previous observations on codon effect, also potentially during proliferation, and as well provide a mechanism explaining the effect of age on mRNA translation through cellular energy. There are probably more consequences to investigate, such as other situations where the energy metabolism is abnormal, like in cancer. So let's see what we find with that further. With this, I'd like to thank you for your attention and mention everyone who's working on this. The papers are here, and this is a link to this preprint about codons. Thank you.
Thank you so much for a great talk, and I'm sure there's a lot of questions I know I asked you, but let others ask first.
Hi, great talk. So you highlight ATP. So in other ways, or in other words, you're going to talk about amino isolation as being rate limiting. Do you understand what I'm saying?
Not really.
Well, I mean ATP.
Yes.
I mean you have to activate the tRNA to isolate it, and that's where you require ATP. In the cycle of elongation and translocation, it's GTP.
Sure. No, so yeah, we look at both ATP and GTP together. The two are in rapid conversions, and so in the equations, it's both ATP and GTP that we are considering. We're not making the distinction.
Okay. Have you measured differences in amino isolation?
No. It is actually very scarce. The data of even in intracellular ATP concentration in humans, there are reviews about that, and the variations are very large. We found in yeast, apparently, or robust measures, but they should be investigated further, I believe. Yes?
Really cool stuff. So along with the energy effect you're showing, I guess you could also see, so codon-mediated decay might depend on translation initiation rate. Like if there's a global decrease in initiation, it looks like the codons affect stability less. And you can have, is there any evidence for things like global changes in EIF2 alpha phosphorylation that are different between these tissues, and maybe even the same thing with initiation changes with EIF2 alpha and elongation changes with EEF2 phosphorylation that could also sort of globally change the rate of elongation and make things more prone to degradation in some tissues. I have no idea if that could happen.
Certainly, yeah. There's a lot of overlapping pathways in these things. We looked at the elongation cycle. We have an hypothesis focusing on the elongation cycle and the loading of the ternary complex. There can be also initiation, as you mentioned, playing there. We have not looked at it. It's also, I would say, even not clear whether our experiment in yeast with this very rapid check is rapid enough to entangle those two. So we certainly need more experiments in this direction to see what the exact role is. But one possible explanation is this biophysical model that is focused on elongation. Doesn't mean other mechanisms are not at play.
So I'll ask on the first part, just to balance things, with two somewhat related questions. So one is, you started from saying, let's predict splicing, and you showed some beautiful work you did there. Then you said, I'm going to apply it for genetic variants. You said, look, when I actually know where the splice sites are, because I probe across GPEX, I get much better performance, and you show the precision curves go up. And then if I have a cat that I find against the tissues, and I do this, I go way up, right? So this is great. If I was a clinician, I would say I'd flip it, given what you're saying, right? So if I have a patient that it's undiagnosed, the first thing, based on what you're showing me, that I'll do is I'll sequence it with CAT. It's cheap, it's easy nowadays. And then the question is, what would have happened if you flipped your order? How much of a gain would these deep learning models give you compared to the CAT?
So I'm very lucky because it's on the curve. Thanks. Yes, one could argue let's do RNA first and then DNA.
Yeah, become DNA first.
And
RNA first, exactly.
It is very rarely done, but I agree that this is a nice one. And this is this yellow curve. So that's just the RNA. You only call aberrant splicing from the CAT, from the transcriptome, and you get this performance. So you're better actually than the DNA alone. And now this adding the DNA to that model, I go from the yellow to the brown.
Right. So the related question is, if you look at what are those cases that you're getting a boost from deep learning models? I was hoping to see this, right?
So I think we've done that in the supplement. So it happens to be in those genes that are not expressed in the CAT.
Okay, so it's
pretty.
So it's where CAT as a CAT fits.
Yes.